Everyone. Thank you for coming in this early. It's also strange for me. I'm not really much of a mornings person, and starting work at 8 a.m. it's pretty stressful for me. So yeah, I appreciate like all of you coming here this early to listen to this lecture. So just a bit about what we're going to be kind of talking about both today and tomorrow kind of morning. We'll be going over coalescent theory for analysis of molecular data. So we'll be basically, today we can sequence DNA. There's one more person we need to admit, here we go. So again, today DNA sequence data is everywhere, and then we can use DNA sequence data to answer a lot of our biological questions, whether that be in molecular ecology or evolutionary biology, genomics, throughout the biological sciences, we can use DNA sequence data. But when we're actually testing hypotheses about DNA sequence data, about variation in our DNA sequences, we need a certain starting point. So again, for any comparison, what we actually do is if we know our expectations, so if we have genetic variation, if we have variation along a DNA sequence, just calculating that variation or summarizing that variation is not enough. We need an expectation. We have to have an expectation going forward so that we can actually understand what that variation that we observe in our samples actually means. And coalescent theory actually tells us that. So the basis of coalescent theory and what coalescent theory really does great is it gives our expectations for how we should, how DNA variation through evolutionary history should come about. So it's really this fundamental theory in the heart of analyzing both genomic data and DNA sequence data. And hopefully within these 2 days, I'll be able to touch upon some basics of coalescent theory. So, and as you will see, some of the most known statistics and some of the most kind of known practices you will see that actually come directly from this theory. So let's kind of start. And let's first start by understanding something about DNA sequence variation and some of the methods that we use to summarize DNA sequence data. So first I'm going to start with just basic definitions. So we're going to kind of talk about nomenclature, sites versus locus. This is important because we'll be using site, we'll be using locus, we'll be using these nomenclatures a lot, and they sometimes also get mixed up. So it's a good idea to kind of define them clearly before we before we start. They also come up a lot in the literature, and sometimes they're used interchangeably, so they can actually get mixed up. So what do we mean by a site? So a site is basically a kind of single nucleotide, okay? A site is a single nucleotide. So a site is a single nucleotide and this is important, and is a locus, okay? So what do we mean by a locus? A locus just basically means a position along the chromosome or position along our genome. So basically a site, a single nucleotide, is also a position along the chromosome. And so a site is a single nucleotide and is also a locus, but a locus is not always a site, okay? So that's kind of the important thing. Every site is a locus. So even if we're talking about one single site, we can call that a locus. But a locus is not always a site. So what do we mean by this? So let's look at kind of an example where we have DNA, and let's say this is a stretch of DNA. And within this stretch of DNA, let's say we have 3 sites with SNPs or polymorphisms, like a CT site, an AC site, and a GA site, okay? And in this sense, when we kind of have this DNA strand, this thing here, for example, this region here, let's say this is chromosome 11, position something like 9,876. This is a site and a locus. We can call this a locus. This is a site or a locus, or let's say not or and, but an or a locus. So we could call this thing a site or a locus. But you know, instead of just concentrating on this one site, on this one polymorphic site, we might, you know, we might do that. This could be like an interesting mutation. This could be a mutation, perhaps that changes the function, is the causative mutation that changes the function of a protein, or, you know, makes it that a polypeptide is synthesized in a different kind of way. So it could be an important mutation. So this site or this locus could be of interest. But you know, we might not only want to study this site, we might perhaps just want to study all of this region here. So instead of concentrating on just free sites, we might, you know, want to kind of summarize variation along this whole region. And we can say, like, for example, we want to analyze a region on chromosome 11 that spans between, let's say, position 9876 to, I don't know, 10784. So when we talk about this region here, so when we're actually trying to analyze DNA sequences between these positions, now this is not a site, but is a locus. Is a locus, okay? So basically we can use locus in two ways. A locus could, you know, kind of be kind of describing one site, or a locus could be kind of describing a larger area or a larger, you know, area along our genome. So again, I make this distinction because even in the literature, you know, sometimes locus means a single site, sometimes it means a range, but a site is always a site, and a locus can be both. So that's kind of like getting things out of the way. But let's now return to our problem of actually summarizing genetic data. And we're going to talk about free statistics that actually summarize DNA variation. So let's okay, let me just put that here. So let's go here and let's talk about summarizing DNA sequence data, okay? So let's talk about some of the ways that we can summarize DNA sequence data, okay? All right. Okay, now there's actually, we have free statistics that some of you, I'm pretty sure that some of you that work on molecular biology or molecular ecology will have heard before. So one of the most frequent statistics is number of segregating sites. Okay, so this is one way of summarizing DNA sequence data. It's usually denoted by S. And another one is average pairwise nucleotide differences. And this is usually denoted by pi, so the Latin, from the Latin alphabet. And the third kind of way of summarizing.
Sorry.
Yes.
You can adjust your camera a bit more closely.
I can try maybe. Is that good?
For me, yes. Is it good for everyone? I guess it's okay.
Well, I mean, you have to enlarge me to. Can you see Meric?
Yes, I can see.
Okay.
There is no any warning in the chat.
Okay. All right. So should I just continue?
Yes. Okay.
Okay. All right. So again, we were talking about free statistics for summarizing DNA sequence data. One of them is the number of segregating sites. The other one is average pairwise nucleotide differences. So that's S, that's pi. I'm pretty sure some of you might have heard about it. And the third one is the site frequency spectrum, okay? Which is called the SFS, okay? So these are kind of the general ways, these are the free statistics that we can actually use for summarizing DNA sequence data along a stretch, like basically along the whole genome or across a stretch of the genome. So now let's kind of define these very quickly, because we'll be working on these statistics a lot. So throughout this kind of course, we'll be using these statistics. So let's come up with an example and let's kind of calculate these statistics together to understand what they actually are and what they actually mean. So let's come up with so I'll just quickly draw an example here. So let's say we have individuals. Okay, so here we have kind of our individuals, and let's say we have 3 diploid individuals. So we'll just quickly do 3 diploid individuals. Say T, C, oops, that's a G, C, C, A. Okay. So since these are diploid individuals, we're going to have, each individual is going to have 2 DNA copies. So I'll just quickly write those like that. Okay, and here we'll have individual 2. And this can have a sequence like this. T, G, C, C, T. And let's go like that. A, C, G, C, G, T. And our third individual, let's say we have a sequence like this, which is A here, T here, T here, G here, C, G, A. And again, T, T, G, C, C, A, okay? So here we have like 3 individuals, 3 diploid individuals. And of course, if we have 3 diploid individuals, we have 6 gene copies. So we can look at here that we have a data set of 3 individuals and 6 gene copies, okay? So when I mean gene copies, so when we talk about, so generally in population genetics, when we talk about gene copies, we don't really actually mean that this region that we're analyzing has to be a gene. It's any stretch of DNA. They're just called gene copies. You can think of these as DNA copies as well. So actually, if we have 3 individuals, 3 diploid individuals, we have actually 6 DNA copies. And when we actually look around here, so I need that space, 6 gene copies. When we look along this region, so we can think about every one of these as a site. So let's also define this region. We can say that this region here is 2,000 base pairs. So when we write things like this, we're saying that this is a stretch of DNA, which is about 2,000 base pairs, and we're only actually showing the polymorphic site. So everywhere else, then the kind of base pairs or the bases that we're showing are actually they're not different. So every single site here is the same in all of those individuals. Every single site here is the same. So we have a stretch of 2,000 base pair regions with some polymorphism around it. And we can actually label these sites. So we can say this is site 1, site 2, site 3, site 4, site 5, and site 6. And as you can see, this individual here has AA, and this individual is TA, this individual is AT. So when we look at this site, we can say that this site is, for example, segregating for A and T. So this is a polymorphic site with A and T, okay? And if you look here, we can say that this is actually a polymorphic site for TC. So there's the T allele and the C allele, and this region here has the G allele and the A allele, and this has, the C allele, T allele. So all of these 6 sites are actually polymorphic sites. And this brings us to kind of, and we can actually let's label also our gene copies like A, B, C, D, E, F. So we have about 6 gene copies and 3 diploid individuals. Now let's try to calculate our first statistics in this kind of sense. So what's our first statistics? Our first statistics is S. So this means number of segregating sites. Well, the number of segregating sites is the most simple statistics to calculate because it's just that. If we go along this 2,000 base pair regions for all of these individuals, how many sites do we have that has polymorphisms? So I'll just admit one more person. So how many sites here do we have that are polymorphic? Well, we have 2,000 sites along this stretch of DNA, and we have 1, 2, 3, 4, 5, 6 regions which are polymorphic or are segregating in the population. So segregation just means that different alleles have to be segregating at that site. So how many sites do we have with segregating allele frequencies or segregating alleles? We have 6. So basically, number of segregating sites in this instant is 6. Or a much more better way of doing it is 6 over 2,000 to get this in per site. So we could say that, for example, this is 0.03 segregating sites per site. Okay, so this is kind of the way that we can calculate this very simple statistics. Now, here's one question for you. So segregating sites just means counting the number of polymorphic sites. Now let's do something. So let's ask a question. What would happen to S if we increase the number of gene copies, okay, or individuals? Okay, so here's a question for you. So the question is, what happens? So here I have my S is 6, so number of segregating sites in this example is 6. I have 3 individuals, 6 gene copies. So what would happen to the statistics if I were to increase the number of individuals or the number of gene copies in my sample? What do you think would happen to S? How would it change? Any ideas? Manos Stuck is saying it will increase. Yes, it will increase. Will it decrease? Can it decrease? Do you think that it, like if we add more individuals, do you think S could ever decrease? I'm trying to read the chat. No, again, no, that is pretty true. Why can't it decrease? Because we already have 6 segregating sites, right? The more individuals only will increase the probability of even getting more sites, other additional sites, which could actually have segregating alleles. So again, S always increases. So the kind of story here is that S increases with, with, let's say, sample number, okay? Sample number. So S can only go up if we increase the number of samples. So this actually means that S is also kind of has to do a lot with the number of gene copies. So if you're actually comparing 2 populations using S, it doesn't really make sense if one of them has a whole lot more sampling than the other one. We need to be careful. And we'll also see that, ways that we could actually fix for that later. So that's kind of our first important statistics, S, which is just the number of segregating sites. Now let's concentrate on another statistic. So instead of S, let's go to kind of our second statistics. So I'll just get these out of the way. Like that. So let's concentrate on pi, okay? So now let's concentrate on our second statistics, so which is, pi, which is average pairwise nucleotide differences, okay? So let's look at how we can actually calculate this statistics and what it actually looks like. So what is average pairwise nucleotide differences? For those who don't actually know. So it's exactly what we're talking about. So here we have about 6 gene copies, and average pairwise nucleotide differences is just a question. If I were to look along these 6 gene copies, what's the average differences between any random pair that I choose from here? So the calculation of this statistics is very simple. Here we have gene copy comparison. So we need to kind of compare our gene copies, and let's say here we have the difference. Okay, and here we have kind of the difference. So gene copy comparison. So we have to actually compare all gene copies with each other, get their differences, and then take the average. So that means I have to compare A with B, A with C, A with D, A with E, A with F, and then B with C, B with D, B with D, B with F, and then just go through the whole thing. So basically what we're doing is, what's the number of differences between A and B, gene copy A and B, A and C, A and D, A and E, and so we can just go down all the way to, let's say, E and F, okay? So this is kind of what we have to do, and we can just calculate this very simply. So what's the number of differences between A and B? Well, I can just look at the sites. First site, I have no difference. So I have one difference here. This is a T, this is a C. I have another difference here. This is an A and this is a G and this is an A. So that's two differences. I have I have a C and T. So that's 3. Here we have a C, here we have a G. So that's 4. And here we have A. So again, between A and B, we have 4. And I can do the same thing for A and C. Okay, I have one difference here, no difference here, no difference here, no difference here, no difference here, and another one there. So that's 2 differences. And I can just like do this comparison all the way through. I can check for, you know, A and D, that will give me 3. I can check for A and E, that will give me 1, all the way down to making all of those comparisons. So that's actually 2. So once I actually have this, all I have to do is take the average of it. So what we're talking about pi is, we're talking about, you know, the average. So pi, to calculate pi, that's just average of this system. So that's just the average of the system. So how do I take the average? It's just simple, 4 plus 2 plus 3, you know, plus 1, blah, 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 all the way to plus 2 over. So what do I divide by this? So what's kind of the denominator here? What would be the denominator to take the average of all of these pairwise differences? Okay, so again, the denominator here is it the number of gene copies? No, it's actually the number of comparisons. So I need to divide this by the number of comparisons, okay? So the number of comparisons, and the number of comparisons here is, of course, 15. So number of comparisons, let me just do it like this. You know, no comparisons, which would be 15, okay? So we could divide that by 15. So this total would be 43, this would be 15, and this would give us about 2.87. So this would be pi. So now we've actually calculated pi for this for this, you know, short stretch of DNA that we have here, okay? And note the difference between, so S was, so let's write S here, S was 6, pi is 2.7. So again, this actually shows that S, since it's just counting the number of segregating sites, always has to be a whole number, but pi doesn't actually have to be. It could be a decimal number as well. Now, we could also, so of course, write pi as, you know, per site. So we could do 2.87 over 2000, which would give us something like 0.001435, and this would be, you know, per site. This would be the per site estimate of pi. So here's the same question that we did with actually S. So what do you think would happen if I were to increase the number of gene copies or increase the number of individuals that I'm analyzing? Would pi go up or down? Would, you know, just like S, would pi increase with the more number of gene copies that I added? Anyone have an idea about that? Okay, so the thing is, pi would actually not increase or decrease with the number of added new gene copies, because pi is an average. So think about this way: I added another gene copy, you know, maybe, you know, with, you know, A, like with this one or with any other ones, that, you know, gene copy is going to have, you know, it's going to be very similar to one of these. So what I'm adding is, perhaps I'm adding, you know, a comparison that is very low in differences. And some of the times I'm going to add another gene copy that has a high difference between any other gene copy. So some of the time this, you know, average is going to go slightly up. Some of the time that I'm adding new gene copies is going to go down. But since it's an average, it's basically going to stay around that mean. So it's going to converge more and more towards the mean. So here, pi does not, you know, change very much with increased sample size, okay? And that kind of makes sense because we're calculating a mean. Again, some gene copies are going to increase. They're going to have, you know, they're going to have more differences with the other gene copies. Some gene copies are going to have less differences. So on average, so that average is not really going to change. So again, pi is kind of more invulnerable to the number of gene copies that we have in this system, okay? So that's pi. So now let's move on to our last kind of summary statistics, which, you know, today is again, perhaps the most important summary statistics that we actually have in genomics, because again, it's a much more, let's say, broader statistics than S and pi, and we'll actually see why when we're actually doing it. So let's move on. So I'm just erasing these. Okay, so let's move on to kind of our last last statistics, which is the site frequency spectra. So site frequency spectra. And we can look at this statistics in two ways. So we can first do this as the minor allele spectrum, okay? So what do we mean by the minor allele? So again, here we're classifying something as a minor, and that actually means that we also have kind of a major. Again, we'll be working with this system here where we have 3 diploid individuals, 6 gene copies. So what's a minor allele? What's a major allele for those who actually don't know? So the minor allele is, let's say here we have, in this first slide, we have 2 alleles. We have the A allele and we have the T allele. Well, the minor allele is the allele which is, has, less numbers, which occurs less. So if we look here, we have 1, 2, 3, 4 A's. So we have 4 A alleles, and we have 1, 2 T alleles. So in this case, T would be the minor allele, A would be the major allele. If we look at this second side here, we can see that we have 1, 2, 3, 4 counts of the T allele, 2 counts of the C allele. So here, C would be the minor allele, T would be the major allele. Here, if we look here, we have 1, 2, 3, 4, 5 counts of the G, 1 count of A. So here, A would be the minor allele, G would be the major allele. So we can actually, for any site, we can go along this site and classify alleles as major and minor, okay? So that's kind of basically it. And we can actually look at this fifth site where we have 1, 2, 3 counts of C and 1, 2, 3 counts of G. So those are both the same. Both G and C are in the same numbers within the site. And in this case, it's not really important. We can treat any one of those as the major or minor allele. So in that, it's just choosing this as the minor one or this as the minor one doesn't really matter because their frequency is the same. So what the site frequency spectra actually does is it classifies kind of these sites according to the, it classifies sites according to how many number of sites there are that has a certain type of minor allele frequency. So when we look at the system, we could have sites that have 0 copy of the minor allele. So that means non-polymorphic sites. There is no minor allele, 0 copy, sites, number of sites with 0 copy of the minor allele. We could have, sites that have only 1 copy of the minor allele. Like for example, like this site here is a site which has only 1 copy of the minor allele. We could have sites that have 2 copies of the minor allele, and we can have sites that have 3 copies of the minor allele. And we can't really have any more sites that have more copies of the minor allele. Like we can't say that there's a site that has 4 copies of the minor allele because 4 out of 6, that would just automatically make it a major allele. So when we're talking about the minor allele frequency spectra, we can actually only divide this kind of stretch of DNA into 4 bins where we have those sites which have 0 copy of the minor allele, 1 copy of the minor allele, 2 copy of the minor allele, and 3 copy of the minor allele. And then we can actually count it. So if we look at this 2000 base pair stretch region, we can say that we have 6 sites which are polymorphic, so which have differences between a major and minor allele. They have 2 alleles segregating, but we then have about 1,994 sites that don't have any minor alleles. So all of those alleles are actually the same. Okay, so what we can actually do is we can actually count and we can say that we have 1,994 sites that have 0 copy of the minor allele. Then we can actually look at those sites that have 1 copy of the minor allele. So I can look at this site, we have 2 copies of the minor allele. I can look at this site, again, there's 2 copies of the minor allele. I can look at this site, and this site actually has 1 copy of the minor allele. So I can say, okay, here I can add 1 step here. So I have, I have 1 site. So we can look at this site, which has, again, as you can see, this is T, that's 1 minor allele. So this is, again, another site that has, another, so we have actually 2 sites with 1 copy of the minor allele. And then we can move to this bin, 2 out of 6. So we can look here and we can say, okay, there's 2 counts of T's, 4 counts of A's. So this is a site that has 2 counts of the minor allele. And we can move here. We can say, okay, we have 2 C's and 4 T's. So this is another site that has, 2 counts of the minor allele. So we can move on. That only has 1 minor allele. This only has 1 minor allele. Here we have 3 minor alleles. And here's another site which has, 2 T's and 4 A's, so 2 counts of the minor allele. So we actually have 3 sites that have 2 counts of the minor allele. And we can look at the sites which has 3 counts of the minor allele, and that's only this site here, which has 3 counts of the minor allele. So that's, again, 1. So this here, now instead of a number, so the good part about the minor allele frequency spectrum for summarizing genetic variation along a stretch of DNA is that it doesn't actually reduce that variation into a number. So remember S and pi, we get a number, we get a single number. We have a 2,000 base pair stretch of DNA, and what we've actually done is we've kind of reduced that variation to a single number. S, we've reduced it to the number of segregating sites. In pi, we've reduced that to the average pairwise nucleotide differences between each and every gene copy. But in every instance, we're reducing all of that variation into a single number. But the site frequency spectra actually reduces it to a kind of distribution of variation. So we can actually look at this region and we can say, okay, we have a lot of sites. We have this many sites that are not polymorphic. We have 2 sites which have only 1 copy of the minor allele. We have 3 sites that have 2 copies of the minor allele, and we have about 1 site that has 3 copies of the minor allele. So it's much more rich, much more, it includes much more information. And again, as we move on, we will actually see that there's a lot of things that we can do with the site frequency spectra, and most of the kind of analysis we do today, whether that be selection scanning, FSTs, or demographic modeling, actually depends on the site frequency spectra. But this, again, is the minor allele frequency spectra. But there's also another way of classifying, and once we've done this, then we can give actually a little break once we finish this. So one way we can actually, so this is the minor allele frequency spectra, but another way that we could actually kind of build our site frequency spectra is not by classifying them as, minor, major, but classifying that as ancestral versus derived, okay? So what do we mean by ancestral versus derived? So what we mean by ancestral versus derived is when we look at this variation here, this is an A, this is a T, this site has an ancestral sequence that it actually diverged from, or, that it actually descended from. And the question is, perhaps, once upon a time, all of this region was A, and then, we got a T mutation. So T is the new mutant, and A was actually the ancestral type. Or maybe here, actually, C was the ancestral form, but T was the new mutant type. So there, but how do we know this? How can we understand this? So we need kind of a way of classifying variation as ancestral and derived, and we can think of it like, so I'll just give an example to clear up. So let's say we have a stretch of DNA, which is like ACG, CC, TG, it doesn't really matter. And here, let's say we have, DNA replication with error. So that just basically means mutation. As we know, DNA always replicates with error. There's always going to be some amount of error, and that brings about mutations. And let's say we have A, T, C, we have G, and here, right here in that C, we actually have a T. So let's do that with like a different color. There's a T, there's a C, T, G, okay? So here what we've actually done is, so when we actually look here, so this DNA is replicating, there's an error, there's a mutation, and now, instead of a C, we actually have a T. So this, in this case, is the ancestral version, and this is the derived version, okay? So this is the derived version of that system. So now if we actually knew this history, if we knew our ancestral starting state, we could actually always divide alleles into ancestral versions and derived versions. But how can we actually know this? Again, so how can we know this in real life? So when we have a sample from a population, we can't really look back in time. Or can we? Again, we could do this, for example, today when we're doing experimental evolution with bacteria or whatnot. We could get a bacterial colony, we could freeze it, sequence its genome, basically freeze that colony, and then, let it, we can take other clones from that and just like let them evolve, let's say, for a couple of generations. And then we could actually go back and look at their ancestral sequence, and we could actually look at their new sequences. But when we actually take samples from natural populations, how can we understand whether one allele is ancestral and the other one is derived? How can we make that difference? Anyone have an idea? No, it could just stay the same. Well, of course it could just stay the same. I mean, if you're referring to this, if I'm kind of, or maybe that's something else, okay. But anyway, so how can I know this? How can I divide sequences into, or alleles into ancestral derived? Is there a way of doing this for natural populations? Yes, compare with other species. Yes, compare with an outgroup. That's perfect. So again, yeah, exactly. That's what we can actually do. So where's my marker going? Okay, here we go. So yes, exactly. That's what we could do. We could compare with an outgroup. So in essence, we could build a phylogeny. Okay, so we could look at basically phylogenies. So we could look at phylogenies to actually answer this question. So how does that come about? So how can we actually know? So let me just erase this for now, this portion here. So what we can actually do is if we have a phylogeny, so if we actually know something like this, so if we have, let's say we have A here, we have T here, and T here, and this is species, let's say this is species B, and this is species A, okay? And we actually know that, here, this is the outgroup, and these are kind of like, the populations that we're controlling. So since we have a T in the outgroup, so this would be ancestral, and this would be derived, okay? So if we actually have an outgroup, and we can compare this, our populations along a phylogeny with an outgroup, we could actually determine, not for certain, but we could have a general good idea which is the ancestral state and which is the derived state. So whatever state the outgroup carries will be the ancestral state, and of course, the new state according to that will be the derived state. So using this phylogeny, using kind of like this outgroup comparison, we can actually divide this polymorphism into ancestral versus derived. So we could characterize, if we have an outgroup, we could characterize each and every one of these alleles as derived versus ancestral. And now let's kind of see how that changes things, how that changes kind of our site frequency spectra. So now let's move on to the second type of site frequency spectra that we actually have. We'll just quickly do this, which is, again, site frequency spectra, but this time, instead of the minor allele frequency spectra, we'll be talking about the derived allele spectra. Okay, and so what is the derived allele spectra? This means that we actually have, this time, an outgroup. So let's say we have an outgroup. And for this sequence here, we have an outgroup, and we can say that the outgroup is A, T, G, C, G, T, and let's just number these so that we can follow along. So this is site 1, site 2, site 3, site 4, site 5, and site 6, okay? So now we actually know the ancestral state of these polymorphisms here. And now we can actually, again, build kind of a distribution. Now that we have derived allele frequency spectra, what we'll be counting is the number of derived alleles in a site. So again, a site could have 0 copies of the derived allele within these 6 gene copies. It could have 1 copy of the derived allele. It could have 2 copies, It could have 2 copies of the derived allele. It could have 3 copies of the derived allele, and it could have 4 copies, 5 copies of the derived allele, or it could be made totally up of derived alleles. Okay, so now, as you can see, we have a much more wider kind of spectrum because there's more bins, more different types of sites that we can add if we classify each and every polymorphic site as each and every allele as ancestral or derived. So we can again do this count. So those sites that have 0 copies of, that's again the same. We have 2,000 sites, 6 of them are polymorphic. So that means that about 1,994 of them don't actually have any variation. So we can do that column here. We can again count the number of sites that have 1 copy of the derived allele. So we can go again, we know that, here, let's look at this site. So that's the ancestral allele. So here T is the derived allele. This has 2 copies of T, so that's not it. Again, we can look here, that's T is the ancestral, so C is the derived allele. So we have 2 copies of C. We can look at the third one, so that G is the ancestral, so A is the derived allele. So again, this has 1 copy of the derived allele, so we actually have 1 site now. We have a site that has 1 copy of the derived allele. We can look at 4, which is C is the ancestral, so T is the derived allele. So again, this has 1 copy, again, T, 1 copy of T. So again, we found another site that has, 1 copy of the derived allele. And we can do this. We can look at this here, which is T is the ancestral. So here T is the ancestral. So 1, 2, 3, 4 copies of the derived allele. So again, so we only have 2 sites that have 1 copy of the derived allele. We can do this for 2. So again, we can look at this here. We can see that A is ancestral, T is derived. So here is a site which has 2 copies, of the derived allele. Again, we can look here. T is the ancestral. So we have another site that has 2 copies of the derived allele, like so. And again, we don't have any of the sites that have that. So we can look at sites which have 3 copies of the derived allele. So we can look here, we can look at this site here, 5th site, which is G is the ancestral allele, and there's 3 copies of C, so that there's a site which has 3 copies of the derived allele. And lastly, we can look at this here, which T is the ancestral allele, A is the derived allele, so we have 1 site that has 4 copies of the ancestral allele, okay, or the derived allele. So this is kind of the derived allele frequency spectrum. So we can say this is the DAF, okay, derived allele frequency spectrum. So there's 2 ways that we can do them. And again, here we have a much more, in-depth, so this gives us more information. And the good part about the derived allele frequency is that if we have the derived allele frequency spectra, we could actually fold this to get the minor allele frequency spectra. So what does actually folding mean? So folding means that, we could put this column here, because again, if we don't know which is ancestral, which is derived, those sites have 6 copies of the derived allele, and those sites that have no polymorphism will exactly be the same, because we don't know which is ancestral, which is derived. So we can just like add whatever's in this column to this column. And again, if we have 5 copies of the derived allele, so that's no different from if we're talking about major minor. So here we have if this is major minor, the derived allele here is the major, and we have 1 copy of a minor allele. And here again, we have 1 copy of the derived allele, but 5 copies of the major allele. So again, these 2 bins are the same, so we can add them here, and we can add this bin here. So what this actually means is if we have the derived allele frequency spectra, we can just simply fold it and get a minor allele frequency spectra. So again, this has more information than the previous one. Just to wrap things up, we've just gone over like 3 summary statistics. And how can we actually do this? So the last question that we can actually tackle is how to like define a locus to summarize. Okay, before we go off to the break. So again, we have these free statistics. So how can we analyze things using these free statistics? Well, we could use the whole genome. So we could go along our genome and calculate S, pi, and the site frequency spectra. We could do this for the whole genome, but this isn't, this in itself could be useful. But again, remember, when we're talking about S and pi, we're talking about a single number. So we're summarizing all of that DNA variation down to a single number. And in site frequency spectra, we're summarizing is this allele, as a distribution of allele frequencies. But that's, again, a huge, reduction in information, because we know that different portions, different regions of our genome could actually have different, amounts of variation. So we're losing all of that, and we're just getting a total. So again, that could be, that could be advantageous. We could use that, for example, if we know the distribution of these statistics across the whole genome, perhaps we can compare it with regions of the genome and see, where, these statistics diverge from genome averages. So that's why it's useful. Or we could do this for a specific, locus or gene. Maybe we have a certain gene of interest, and then, we want to kind of determine how, variation is distributed within that specific gene. Or we could do something like a sliding window. Okay, so we could do a sliding window, which is, today in the genomic age, when we're actually analyzing genome-wide data, sliding windows helps a lot. So what is a sliding window? So a sliding window means that we'll be analyzing our genomes window by window and then moving forward. So again, let me just give an example. So let's say we have a window of 10,000 base pairs window. And let's say we have a step size of 5,000 base pairs, step size. So what does this mean? What does this do? So let me just draw it here. Let's say we have a genome like this, and this is our genome. And what we're actually doing is we're walking along this genome in 10,000 base pair kind of window. So we can start here, kind of scanning along our genome in pockets of 10,000. So each of these, so this here would be 10,000 base pairs, and this this portion here would be 5,000 base pairs. So each time we're doing our analysis, and for this, for example, for this window, let's say we're calculating pi. We're calculating pi for this window, then we're moving along 5,000, then we're calculating pi for this window, and then we're moving another 5,000, calculating pi, calculating pi, calculating pi, calculating pi. So we're calculating a bunch of pi's as we're walking along our genome. And what does this help us do? So this, this helps us in understanding how pi changes as we move along the, kind of our chromosomes. So let's say, these are kind of our windows that we're scanning along, and this is pi. So we can actually see, for example, in the first window, pi could be something like this. In the second one, it could drop. 3, it could be like this. Then 4, it could be high. 5, it could be high. 6, it could be. So we could actually, walk along our genome and kind of see how pi changes as we go to different regions of our genome. So again, this is kind of sliding window technique. And we can just not do this for pi, but we could do it for the site frequency spectra. We could see how this distribution of allele frequencies actually changes as we move along the genome. And we can actually compare different regions to see where regions, pi is high, or where genetic variation is high, where genetic variation is low. But here's the question: what does that actually mean? What does that actually mean? And we'll get to that question in a bit. So what does that actually mean? Like pi being high, S being high, or S being low, or the site frequency spectra having a certain shape or having a different shape. What does that mean? How can we give kind of meaning to that? Because these are just numbers, right? So to have a meaning for something, we need an expectation. So if we have, for example, under a certain model, if we have an expectation for these statistics. So for example, you could compare this with the well-known Heidi Weinberg kind of model. What does the Heidi-Weinberg equilibrium say? It says, under a certain circumstances, if you give me allele frequencies, I can predict genotype frequencies. So if there's no population, if we have a population of infinite size, if there's no selection, if there's no migration, if that population is stable, if there's, no differences in, sex ratios or whatever, if all of those assumptions are met, if you give me allele frequencies, I can predict genotype frequencies. I can say, okay, genotype frequencies in the population would look like this. And again, and that is what we actually need here as well. So, we need an expectation for these statistics given a certain model. If we have expectations for how this variation would look across the genome, then we can actually start, making sense of these. We can have pi of a certain number. If we know what to expect pi under a certain model, then we can say, okay, I'm not seeing the same pi. My pi is not it doesn't conform to the predictions of my model. Perhaps some of those assumptions of that model is wrong. For example, if it's a Heidi-Weinberg, model, we can actually say if my genotype frequencies that I observe don't fit the predicted ones, I can say, oh, perhaps this population is under selection, or perhaps this population is receiving migrants. So it gives me information, and it's the same thing for this. And that is where coalescent theory comes. Coalescent theory actually gives us predictions for these statistics. So it predicts the number of segregating sites under a certain model. It predicts pi, it predicts, SFS. So basically it predicts genetic variation along our genome given a certain model. And that's kind of the model we'll be looking at after the break. So we'll be looking at coalescent theory. We'll be building up from the ground up. So we'll be building coalescent theory to see how it actually predicts genetic variation across the genome and how we can use that predictive power to answer some of our important questions. So now let's kind of give a break for about 10 minutes, and then we can actually resume. Major allele, can it be ancestor? Yes, it can be ancestor. So again, in this case, for example, here, if you look at it, which one was it? Yeah, this one here. The question is major allele, can it be ancestor? Yes, of course it can be ancestor. Like in this case, A is the ancestral type, and it's also the major allele, so it can be. So an ancestral allele can be both major or minor, it doesn't matter. So because major and minor just means, major means, you will have more of that allele in the population. The frequency of it will be higher. Minor allele means the allele which has lower frequency. In this case, A is the major allele which has higher frequency, and T is the allele which has a lower frequency. So T is the minor, A is the major, but that has nothing to do with ancestry. Usually ancestral alleles actually are more frequent in the population, but that doesn't always have to hold. So here, as you can see, T is the ancestral allele, A is the derived allele, but A is actually the major allele and T is the minor allele. So this means that, for example, perhaps this mutation here, this new type, since A is ancestral, this A type, maybe it had an advantage, so it actually spread across. So, it increased in frequency in the population. Is it more possible major allele be the ancestral one? Yes, most of the time. So again, most of the time when you have a new mutation, it takes time for that to actually, if it's an advantageous mutation, it might, due to selection, increase in the population, and at some time, transverse, the major allele, become the major allele itself. But again, usually, as you know, mutations, they remain in the population or they get lost very quickly. So usually ancestral alleles are the major alleles if you don't have selection or if you don't have a reason for that new mutant to actually increase in frequency in the population. But there is no one-on-one correlation. Ancestral alleles aren't always the major allele, and derived alleles aren't always the minor allele. That's one power of knowing ancestral derived states, that we can actually understand the system. So if we look at this site, we can actually see that although T was ancestral, this new derived, this new state, this derived allele A has actually become the major allele in the population, and that tells us something about that mutation. It tells us, once upon a time there was a new mutation that actually reached higher frequencies in the population. So again, there's no one-on-one correlation, but all of it just basically gives us more information. Okay. All right. So I'll see you guys. So Meric, the break time is yours when when the break is enough, we can re-meet.